Are you genetically modified? You probably answered no, but you may be thinking, how do scientists modify a genome? Genetic engineering really took off during the 1970s with the development of recombinant DNA technology. Two brilliant scientists by the name of Herbert Boyer and Stanley Cohen were the first people credited with the development of recombinant DNA technology, which showed that genetically engineered DNA molecules may be cloned in foreign cells. Throughout the rest of the 70s, recombinant DNA technology was researched thoroughly and further developed. Eventually, in 2012, the CRISPR genome editing tool was developed, which revolutionized genetic engineering technology. Now, you may be thinking that CRISPR is an artificial technique created by scientists, but CRISPR is actually a naturally occurring defense mechanism found within bacterial cells. In the 1980s, scientists found a phenomenon in the DNA sequence of bacteria. What they saw was a repeated DNA sequence with different segments in between. After further research, scientists discovered that bacteria would hold on to parts of viruses and insert them into their own DNA in order to defend themselves if they were attacked by that same virus again. The defense system utilizes enzymes called Cas, known as CRISPR-associated proteins, which are able to precisely cut DNA. Bacteria use these proteins to snip off parts of viral DNA to insert into its own DNA. Pretty cool, right? Scientists call the unique segments between the repeated sequence spacer DNA, and they call the entire configuration clustered regularly interspaced short palindromic repeats, giving the name CRISPR. But how can scientists use this to help us? CRISPR-Cas9 allows geneticists to modify a genome by modifying the DNA sequence. The CRISPR system can be engineered to edit eukaryotic DNA by designing guide RNA complementary to the target sequence. What this does is tells the Cas9 protein where it should snip the DNA. Once the DNA is cut, researchers use the cell's own DNA repair system to add or delete pieces of genetic material, or to make changes to the DNA by replacing an existing segment with a customized DNA sequence. I know you may be thinking, why is CRISPR so important? Aren't there other simpler ways to modify DNA? You'd think so, but there really aren't. CRISPR is arguably the simplest and most efficient method of genome editing. Additionally, since it can be applied directly in an embryo, CRISPR reduces the time required to modify target genes compared to gene targeting technologies based on the use of embryonic stem cells. Along with all of these advantages to CRISPR, there are still many limitations and further research that must be conducted on CRISPR before it becomes a viable and reliable system of genome editing that can be conducted safely in clinics throughout the world. For one, it is possible that the guide RNA that guides the Cas protein ends up binding to a sequence that it doesn't actually complement, resulting in insertion at a place in the genome that wasn't intended. This can cause problems like cancer to occur in living people treated using CRISPR. With future research on CRISPR and new additions to the procedure, CRISPR will become a safer and more reliable method of gene therapy and genome modification. With the number of research papers being published about CRISPR each day, it is only a matter of time before CRISPR procedures are widespread and used in every clinic throughout the world.